Hello, everyone, and welcome to what promises to be an enlightening fireside chat. This is Paul Kuntz with Redship Companies, and today we're thrilled to have with us two distinguished guests, Tom Chittenden, Chief Scientific Officer of BullFrog AI, and Dr. Daniel Weinberger, Director and CEO of the Lieber Institute for Brain Development. Before we begin today's discussion, I'd like to remind everyone that BullFrog AI is a publicly traded company on NASDAQ under the ticker symbol bfRG. And during today's conversation, forward-looking statements may be made within the meaning of the Private Securities Litigation Reform Act of 1995. Any statements that are not historical facts should be considered forward-looking, and forward-looking statements involve risks and uncertainties. Actual results may differ materially from those discussed here, and we encourage our listeners to refer to the detailed disclosures in BullFrog AI's filings with the SEC for further information. We will be opening the floor for a live Q&A session following the initial discussion with Tom and Dr. Weinberger, and that will give you the opportunity to engage directly with our guests. Now, to kick things off, we're going to play a short video from BullFrog AI founder and CEO, Vin Singh.
Thank you for joining us today for this momentous occasion. I'm Vin Singh, CEO of BullFrog AI, and it is my privilege to welcome you to our virtual presentation. Today, we will delve into the groundbreaking advancements resulting from our strategic collaboration with the Lieber Institute for Brain Development. Let me set the stage. BullFrog AI, a technology-enabled drug development company, has harnessed the power of artificial intelligence and machine learning to drive precision medicine. Our mission is to accelerate the discovery and development of pharmaceuticals and biologics that can transform lives. Now let's talk about Lieber. As a nonprofit research institute, Lieber maintains the world's largest repository of postmortem brains dedicated to the study of neuropsychiatric disorders. Their comprehensive brain data, genomic and other data, provides an invaluable resource for our collaborative efforts. So what have we achieved together? So far, we've uncovered hidden relationships. Using our proprietary bfLEAP AI platform and causal AI, we've mined Lieber's data to reveal previously unrecognized connections between genes, pathways, and brain disorders. Imagine the potential impact on treatments for conditions like major depressive disorder, schizophrenia, and bipolar disorder. We've also discovered novel drug targets, and we continue to do this work. Our collaboration is leading to the identification of novel drug targets, which is really the first step in drug discovery. These may hold immense promise for treating a range of debilitating neuropsychiatric disorders. Our objective of this partnership is to engage with pharma with a goal of securing multiple target discovery deals, and we are currently doing that. Any deal that we secure would likely represent transformational revenue and profits for BullFrog AI. In the coming minutes, you'll hear from Dr. Daniel Weinberger, CEO of the Lieber Institute, and Dr. Tom Chittenden, Chief Science Officer for BullFrog AI. They'll delve deeper into our approach, findings, share insights, and discuss the transformative potential of our collaboration. So without further ado, let's embark on this exciting journey at the intersection of AI, neuroscience, and hope. Welcome to the BullFrog AI Lieber partnership, a beacon of vast potential in neuropsychiatric research.
Okay, gentlemen, we will get things started here. Again, I want to thank you both for joining us today to share your insights on this transformative collaboration. Tom, we'll kick things off with you. Since the announcement of your partnership, what have been the most significant milestones or achievements, and could you highlight any particular breakthrough that stands out?
Absolutely, Paul. Before we get started, I would like to thank you for the opportunity to share the work that we have engaged in with Dan Weinberger's team at the Lieber Institute. And I think it's also important to recognize what I'm going to be sharing or discussing here this afternoon. The lion's share of this work has been generated by our data science team, which includes Enrique Garcia Rivera, Juan Felipe Beltran, and Jeremiah Walla. They are just three brilliant data scientists, and the data or the results that I'm going to share with you today are just absolutely spectacular, to say the very least. So as far as the achievements go, I've been here 4 months, and when I started at BullFrog, we were working with some proprietary software, graph-based analytics that the organization had leased from the Applied Physics Laboratory at Johns Hopkins. And we were working very closely with Chetan Salvki, one of the chief AI advisors for BullFrog. And so what we have done from an AI or from a platform standpoint is that we've supplemented this graph-based analytics with something called generative and causal AI. And so what we have done over the past 4 months or so is that we've analyzed the entire data set at the Lieber Institute. And so this includes 2,800 brain samples across 5 brain regions and 3 neuropsychic disorders, including schizophrenia, major depressive disorder, and bipolar disorder. So Enrique's team started with the analysis, after the pre-processing of the data, harmonization, so that the results aren't based on some type of technical artifact. They ran this unsupervised approach to first classify the data. And what they were able to do is generate 68 novel or unique patient clusters based on gene expression profiles. These are very unique clusters, and since then, we have looked at the gene signatures or the patterns within the data that actually drive these patient clusters. And now we're applying something called causal AI to build gene networks or causal dependency structures that indicate 2 or 3 genes that may be driving the state of all of these other patterns within these clusters. And so I think that provides enough overview here for a very in-depth conversation with Dan and what he's done and what he's bringing to the table with Lieber.
Thank you, Tom. And Dr. Weinberger, can you share your perspective on how the partnership between BullFrog AI and the Lieber Institute came about, and what were the initial goals and motivations behind this collaboration?
Yeah, so let me give you a little background of how we got together with BullFrog. So the Lieber Institute for Brain Development and the Maltz Research Laboratories was a philanthropically founded institution here on the Johns Hopkins Medical Campus. We are an independent, not-profit 501c3 institution trying to understand the basic mechanisms in the human brain of the very illnesses that Tom talked about: schizophrenia, bipolar disorder, depression. Also, we study PTSD and some neurodegenerative disorders. But those 3 illnesses were the main focus of the effort here to use the latest technology in understanding how genes are regulated in human brains to translate into these illnesses. And to do that sort of work, it's a no-brainer. If you want to study how the brain is a factor in these conditions, you have to study the brain. And you can't study living brain at a molecular level, but you can study brains from people that died with these illnesses. And the way we've invested in that for the 13 years that we've been here is to collect brain donations from next of kin of people who die unexpectedly with these conditions. And these brains come principally from medical examiners' offices, 4 medical examiners' offices around the country. We get donations of over 400 brains a year. And as Tom mentioned, we have brain data now on thousands of human brains. And these data are very highly complicated, we call high dimensional data, of how genes are turned on and off in the brain, in the human brain. And genes are the mechanisms by which brains grow, they are healthy, and they become sick. And we know that genes are the major risk factors in all of these psychiatric conditions, and how these genes regulate development and function of the human brain is the secret to how these brains manifest these conditions. So over the past 13 years, we've generated a huge amount of data in what genes are turned on, what genes are turned off in different parts of the brain by actually measuring the expression of these genes in these brain tissues. This is very highly complicated data. It not only involves many people, it's multiple brain regions. There are 20,000 genes that we can identify in every individual's brains. And these genes have many different features that have to do with how the genes are processed in the brain that build in even more complexity. And we've spent a lot of time analyzing these tissues, these brains, using traditional technologies, standard statistical methods, what we call general linear model methods of how illness relates to differential abundance of one gene or another gene, or how genes may talk to each other. But it's pretty, it's pretty basic stuff. And we've realized for a number of years now that there's much too much data to be able to use these sort of off-the-shelf technologies that are traditionally used to analyze the abundance of a gene, how a gene correlates with another gene. These are very garden variety strategies. The data are much too complicated. The other issue is that nobody's sick because of one gene or another gene. You're sick because of the cells in your brain are processing a great deal of information abnormally. And that processing is because of networks of genes, not just one gene or another gene, but networks of genes that interact with each other and work together to translate into how the cells work in your brain. And there's no question that strategies that don't start with one gene or another gene, but says, let's look at the whole catalog of how genes are regulated in the brain in a much higher dimensional way, requires data-driven strategies, machine learning, artificial intelligence. We at the Lieber Institute for Brain Development have had a relationship for a number of years with the Applied Physics Lab at Hopkins, which is a uniquely powerful data-driven science group. And when we heard about there was a spin-off basically of technology from the applied physics in this new company, we were engaged in a conversation with them around the idea that we have so much data on so many different dimensions, so many different opportunities to explore these data with novel strategies for how to pick out critical signals from a very crowded room of signals. This seemed like an ideal strategy to explore these data. And we started a conversation with BullFrog, got a sense of what their technology could conceivably do to this is a needle in a haystack, looking for a targetable therapeutic based on very high dimensional data analysis, and a therapeutic that had never been imagined before, but that is somehow lurking in these data. Is the potential of artificial intelligence. And so we were excited about the idea that we could share these data with BullFrog in a basically exclusive relationship, at least for a period of time, and they would explore these data using their tools. So that's what really generated excitement from the Lieber Institute. Our goal is to use this highly complicated molecular data to identify the mechanisms of illness that are treatable. That's the idea of a therapeutic target. And so as Tom mentioned, some of the preliminary work, we still have a lot of work to do, but some of the preliminary work has pretty much blown us away. I mean, we did not expect some of the results to look as interesting, provocative, potentially transformative as some of the work has to date.
Excellent, Dr. Weinberg. Thank you. And that sets up our next question very well. Tom, how has BullFrog AI's platform specifically contributed to the advances in understanding neuropsychiatric disorders? And can you provide examples of the insights gained through AI that were not possible before?
Yeah, great question, Paul. And I think to get back to Danny made a statement about the high dimensionality or the complexity of the data that we're analyzing. That has has caused problems in the field for over 20 years now. So the multicollinearity, the feature dependency, the correlation bias that exists makes these algorithms non-reproducible or unreproducible, if you will. And so what we have done from a technology platform standpoint is that we have addressed all of those issues. And I think time is going to tell how much we've advanced the neuropsychiatric space here. We're very early, like like Dan has mentioned, very early on with this analysis. But what I'm very interested in is what Enrique and his team has uncovered with this unsupervised approach of generating these 68 patient clusters based on these gene expression patterns. And so let me unpack that a little bit here for you. A traditional way would just ask the algorithm to find patterns based on the differential diagnosis, schizophrenia, major depressive disorder, and bipolar disorder, and the algorithms will find those patterns. But the approach that we had taken is an unsupervised approach where we strip off all of those class labels and we let the algorithms cluster the data based on patterns, data-driven patterns within the data. And so what this is pointing to, and again, the operative word here is pointing to, because we have a much more work to do, but it could be an indication of molecular subtypes, say, for example, in schizophrenia. So what was surprising to us is that all the schizophrenic patients didn't cluster in one patient cluster. There were multiple schizophrenic patient clusters that again could represent potential molecular subtypes that we would have completely missed if we had taken more of a historical approach to classifying the data from a supervised strategy there. We would have completely missed all of that. And so now what we're doing is we're pulling out the genes or those gene patterns that are responsible for those clusters, and then applying causal AI to them to see if we can get at drivers of potentially, driving disease, if you will. Now, these tools are perfectly suited for identifying potential drug targets. And that, from an economic standpoint and from a business model standpoint for BullFrog is what we are talking with these pharmaceutical companies about. Are there novel drivers that we have uncovered with this novel approach of analyzing the data, clustering the patients in a unique way, and that has the possibility of uncovering possible or putative drug targets and drivers of disease.
Excellent, Tom. Thank you. And similarly, Dr. Weinberger, from your perspective, how do you anticipate the collaboration with BullFrog AI will enhance Lieber's ongoing research efforts in understanding neuropsychiatric disorders?
So again, the Lieber Institute is all devoted to identifying the mechanisms of these illnesses. There is no treatment in psychiatry today, no treatment, whether it be for schizophrenia, bipolar disorder, depression, PTSD, obsessive compulsive disorder, anxiety disorders. There is no treatment that was discovered based on any basic understanding of the causes of these illnesses or the mechanisms of these illnesses. Every medicine used, and it's true not just of medicines, it's true of magnetization treatments, electricity treatments, talk treatments. None of the treatments are based on understanding of basic mechanisms of illness. Now that we have this kind of molecular and genetic capacity to explore these illnesses in this way, we are going to identify basic mechanisms of illness. And because these illnesses are about the biology of these cells. So that is a strategy for identifying treatments based on causes. It also, and I think the other point that Tom made, is by having all these different clusters with different patients are represented in different clusters. This is the holy grail right now of what we call personalized medicine, precision medicine. We know that one size does not fit all for a particular treatment. And this is particularly true in psychiatry, where there's no treatment, even when it's effective, is not effective for everybody. And it's not effective for a significant percent of people. It's not like 2% or 5%, it's 20, 30% of people with a typical diagnosis do not show good response to what the best treatments are. Sometimes it may be 40 or 50%. So clearly, these are disorders that have multiple different mechanisms, etiologies, causations. And so these clusters, which identify different subpopulations of patients, we believe hold clues to what individualized treatments would be. that would be what this, this whole era of so-called precision medicine is meant to accomplish. So from the point of view of the Lieber Institute, we exist to find new therapies. We exist to find treatments based on causative mechanisms of illness and to use those treatments to reduce this burden of illness in populations, with the expectation that one size won't fit all. And the more we can dissect out discrete pathological mechanisms, the more we will be able to personalize medicine. So this, whatever discoveries come from this are, harmonize beautifully with what the purpose of the Lieber Institute for Brain Development is.
Excellent. Thank you, Dr. Weinberger. And Tom, you mentioned unsupervised models earlier. Can you explain the process and importance of the unsupervised construction of graphical models in this research, and how do these models help in identifying new biomarkers and drug targets?
Yeah, Paul, and I think I've explained a little bit of that in the two earlier questions that you posed, but I've been working in this field for 20 plus years now, and one of the things that have plagued the machine learning space here is the irreproducibility of these algorithms. Is that you can find a signal in a specific patient cohort, but you go to a second patient cohort and that signal's not there. So you're not capturing, what that is an indication of is that you're not capturing true biological variation that's underlying a lot of these disease mechanisms that Dan alluded to earlier. So I've never trusted a single run of a single algorithm on a single partition of data. And what we've done here at BullFrog is that we are running multiple partitions of that data. So as many as 1,000 partitions of that data. These are probabilistic models. So you expect to get a bit different answer with each run of these data sets. And what we are finding, the stability of these clusters are just absolutely, and please forgive the pun here, mind-blowing, is that the very, very reproducible. And so to step back, the difference between unsupervised or generative AI versus that and supervised AI, we're running a combination of both, but we're stripping off the class labels, as I had mentioned earlier, and we're letting the AI go in and find these patterns. And based on these aberrant patterns of gene expression, it's clustering the patients very uniquely. And that sets the stage for moving downstream to looking at the drivers or the patterns in these gene expressions that we are looking at, and then setting the stage for causal inference on top of that. So it's a combination of both unsupervised and supervised analysis that at the end of the day, as Dan had mentioned earlier, we're taking a systems approach, a network biology approach that I believe is second to none. And that is truly the difference between, what our competitors are doing and what we're doing and what we're bringing to the table as far as this partnership.
Great. Thank you, Tom. And Dr. Weinberger, how do you foresee the integration of your findings into clinical practice? So what steps are necessary to transition from research to real-world application in treating brain disorders?
So again, this is drug development 101, so to speak. What's unique here is the strategy being used to find the targets. So drug development starts with a target. You have to have a target that you think will yield therapeutic effects. So what BullFrog is doing with our data is applying we believe, novel and potentially, I think, very informative way of having targets emerge from the data without us pre-assuming that we know what the best targets are. But then the next step is, so if we have a target that we believe using BullFrog technology is a driver gene in these clusters that are relevant to some patient with some group of patient with some particular diagnosis, we have to do something, and we think that driver gene is what we call druggable. We have to do something to validate that that really represents a critical player in the network of genes that these clusters illustrate. So we have to do some experimental biology, and in our collaboration with BullFrog, that's something we have on the table as something that would follow up to the identification of what this target might be. So that's, that's laboratory work that can be done in a lot of different ways. Obviously, animals that can be created that have that particular target or don't, and that we can see how that affects the behavior or the function of the brain of animals. We have a lot of living cells that are stem cell models that we've developed from the actual people whose brain donations we have. So we are able to grow living cells from a portion of the cadavers. And those cells become cells that can be created into little brain models that then can be used to validate what we think is a therapeutic target. And that validation is done with a variety of laboratory experiments to change the expression of the gene of interest and see what happens to the biology of these cells. So we think there's a path to go from having a target to validating that that target really looks therapeutically of interest. And then the next step is to find a small molecule that finds that target and corrects it. And that's that's the drug development world. Once one has that kind of molecule that looks to be a drug that targets this target and that affects the biology of these that are represented in these clusters in a therapeutic way, you have a drug to put in human trials. And once it gets through human trials, it becomes a therapeutic in the clinical marketplace. So there's a number of steps to go from identifying what looks to be a target using this BullFrog technology to having a clinical, a clinical drug. But the path to go to make that happen is a pretty well worked out path. It's not mysterious of how to go from a therapeutic target to having a therapeutic agent. It takes time and it takes some resources to do that.
Excellent. Very good. Thank you, Dr. Weinberger. And Tom, back to you. Could you elaborate on the key findings the collaboration has produced so far, and what do these findings mean for investors?
Yeah, so I think the key findings right now, we're very early on in this collaboration, this strategic partnership we have with the Lieber Institute. But again, the key findings here, and I think of real interest, and I think Dan has expressed this as well, is this unsupervised clustering that we have brought to the table that will allow us to move down that drug discovery, drug development pipeline in an expedited fashion. So we're going to find unique drivers of of these differential diagnoses here, these three clinical indications in neuropsychiatric disorders. We're going to find novel drivers based on these novel clusterings of the data. That sets the stage to move downstream for finding causal drivers with all this. Now, I think it's very important to point out that I'm a real proponent of dispelling the hype that currently exists in the AI space. And I'm getting to your question about why this is important and what does this mean to investors, is that talking with some of my colleagues would love to lead you to believe that we've already cured cancer. We're not there yet. We're getting much closer to that. But what these algorithms do, or the power of AI, is its ability to generate working hypotheses. And so we are at that point in the collaboration with Dan and his team where we have generated working hypotheses. And at this point, it's important to lean in very heavily with the experimental biologist and the translational scientist to understand what these algorithms are actually telling us. And Dan pointed out earlier, some of these are going to be right on, we're going to hit the nail right on the head, and others may be a little bit off. And it's that interaction between this cross-disciplinary approach of working with this data that's going to lead us to the point where we have found something significant, unique and significant, that will attract pharma partners, bring them to the table. And I think that is the first milestone with all of this work that we're doing with the Lieber Institute, is that those contracts, and as Vin pointed out in his opening statement, multiple contracts with pharma partners. And that's what we're working on. And we're in the midst of early discussions with pharma now.
Excellent. Thank you, Tom. And Dr. Weinberger, can you shed any light onto the anticipated timelines for achieving some of the key research milestones in the collaboration?
So, I mean, that's a good question. And I think a lot of that will depend on the partners that BullFrog is able to bring in to help accelerate the work. I mean, I think the job that BullFrog is engaged in identifying what they're calling these driver genes that are responsible for the integrity of these clusters, which are based on many genes working together. That probably is going to be done within a few months, would be my guess. And correct me if I'm wrong on that, Tom, but that work will probably be done. And then the question becomes, how do you move from a target that looks really interesting? And it's worth pointing out, the drug companies that we've talked to, several now, they're actually very interested in this principle, because every drug that these companies are working on are still Me Too molecules. They're based on old ideas about how drugs work in psychiatric illness. And everybody knows that none of these drugs are treating the fundamental mechanisms of illness. So these strategies to use the molecular and genetic information in the human brain with illness is undoubtedly a more promising strategy to find treatments based on causes, which will vary from person to person. So there's a lot of interest in this. And I think the timeline is really going to depend on how impatient the partners that come in to help validate this work are, because the studies done to validate a target can be done in less than a year. You can have enough weight on the scale of validation to believe that you have validated target. And then drug companies are very good at these high throughput screens to find molecules that affect that target. So, this is the kind of work that I think can go from where it is, will be in a few months, to having what we call a lead molecule that targets the target and that looks to be therapeutic in the various models, including animal models, that would be developed, that would then go into a clinical trial. I really think this is a few years of work. This is not 10 years of work. It's probably not even 5 years of work.
Excellent. Thank you, Dr. Weinberger. And Tom, can you talk a little bit about potential revenue opportunities from the partnership and how the collaboration overall aligns with BullFrog AI's development strategy?
Yeah, so I think where this, and I'm going to bring this back to multiple pharma contracts, is that what we are shooting for. We are a drug target discovery, or our business model is founded around drug target discovery, bringing in pharma to play with this in a way that are these multi-million-dollar biobuck deals. And I just point everybody to the news or these recent press releases for other AI companies. Pharma knows these models, and that is who we are going after these types of deals with multiple pharma companies. And I think what's important to reiterate some of what Dan had mentioned earlier is that the key milestones along the way here, that investors that they are looking at BullFrog and our progress, first and foremost would be that initial pharma contract. Target validation, this is going to happen, I believe, within the next year or so. And with these 68 clusters, if there was just a one-to-one relationship, we're going to hand over 68 potential or putative drug targets to Dan and his team to start evaluating. And then where do we go from that? Advancing these targets through a drug development pipeline, as Dan alluded to earlier, and whether that's an internal pipeline here at BullFrog, or we sign a contract where pharma takes these signals and they run with them. And then how do we expand the platform? So this is a foundational model that we are learning a lot with the Lieber Institute. And where we're going with this from an R&D standpoint, because as a CSO, I'm responsible for setting the R&D agenda, is moving to other organ systems. So first, what we're going to do with the Lieber Institute is we're going to build a molecular physiologic map of the human brain. And this is going to be an in silico model. And so what we can do with all of these causal modeling that we do, or that we've generated now, is build an overall network of brain function. And then we can get in and we can ask some very sophisticated questions when we build out potential outcome and counterfactual analysis. We can tweak or change the state of one of these patterns that Dan and his team think that are important or believe to be important. And then we can see how all of that patterning changes throughout the network and associate that with a potential patient outcome, such as disease severity. So that would be the first model that we would go after. But expanding the platform to other organ systems, we're looking at this as well. Pharma has an interest in this, building out other models and expanding our market potential. A model of the human heart, the kidneys, the liver are all areas that we're actually working on. And the lessons learned here with Lieber are going to be very vital to moving things down the road. Earlier, I said that some of my colleagues would lead you to believe that we've already cured cancer. We're not there yet, but I'm quite confident that we are going to get there. Maybe not in my lifetime, but within my 10-year-old granddaughter's lifetime, I'm sure we're going to be able to eradicate disease. And I believe AI is going to be crucial in that. And again, asking the right questions. That's the power of AI. And so if investors are looking at this or listening to us here today, I've just laid out a roadmap for BullFrog that sets a course for us for the next 5 years or so. And within the first year, we are going to be handing over these putative drug targets to Dan and his team to say, hey, do these make any sense? Right?
Yeah.
Excellent. Thank you, Tom.
Sorry, Paul, that was a little long-winded.
Oh, no worries. No, fantastic answer and a very enlightening discussion we're having here today. We are going to now actually open up the event for the Q&A portion. So for our audience, if you do have a question, you can click the Q&A button at the bottom of the screen and just type your questions in. We've had several that have already come in, so I'll just throw a few of these out here. The first is BullFrog has talked about reducing the timeline for drug development. How is this partnership demonstrating that, and what would you estimate the timeline would be for these discoveries without bfLEAP?
Right. I think that's a very important question. So if you look at traditionally what has done in the drug development, drug discovery, drug development space, you can look to the literature and it's an average of 10 to 14 years and $2.5 billion to bring a drug to market. Well, within the first 4 months since I've been here, and we've actually been working on this for 6 months in total, is that we have already defined putative drivers of these actual clusters. And so we're already dramatically decreasing the time in that first step, that foundational step that Dan had mentioned earlier, is identifying efficacious drug targets to move down that discovery and development process with that. So, again, within the next year, or within the next 6 months. So the next time we get together and have a conversation, this may all look like child's play. We may be talking about specific signatures that Dan and his team are already on the way to validating, or if not validated in one way or another.
Excellent. Thank you, Tom. And our next question, my father was a famous research psychiatrist, Dr. William W.K. Zung, and he would be absolutely fascinated with what BullFrog AI is doing where he's still alive today. Are there any qualitative or quantitative results from the last month of AI processing that you can share with us today?
Yeah, Dan, you want to lean in on that one, or
Well,
I can.
I think Tom could probably do a bit, but part of what really has impressed us as neuroscientists, geneticists, brain scientists here at the Lieber Institute for Brain Development is that some of these clusters have a remarkable specificity for dissociating these cases from each other. And so we've seen some clusters where the odds, for example, that you're a bipolar disorder subject rather than any other diagnoses in a cluster could be quite dramatic. Clusters where you have, for example, a 20-fold likelihood that a patient in this cluster has bipolar disorder, as opposed to depression or schizophrenia, or be a normal person and have no psychiatric diagnosis. So those are some of the examples of how this strategy has created molecular subpopulations that are, as you know, as Tom said, these are unsupervised findings that all of a sudden you have a cluster which segregates out a particular group of patients very dramatically compared to all the other patients, suggesting there's something about that cluster that's particularly relevant to that to that patient population. And then the effort to identify causative genes that account for the characteristics of that cluster profile theoretically are a potential therapeutic target for that group of patients that are identified based on that cluster. So that so far has been pretty remarkable for us to see something like that. We've never seen anything like that before, either in any of the work from the Lieber Institute or in any of the literature about the brain biology of psychiatric illness. So that's, you know, that's the kind of, I would have to call that very, you know, titillating kinds of data, because you look at this and you go, wait, this is something nobody's ever seen before. So that's an example.
Very exciting. Thank you, Dr. Weinberger. And our next question, what are the benefits of adding the causal AI into the process?
Yeah, so that is a very relevant question here. Earlier, I had mentioned that we had moved away from, kind of the traditional classification model where we find signatures that associate with disease, to actually integrating probabilistic modeling or probabilistic programming, which we refer to as causal AI, to derive these causal dependency structures that are reflective of the signal transduction cascades that drive cellular behavior and dictate phenotype. So really what we're going after with these models is to model the cause and effect nature of molecular cell biology. And once we're able to define that, then looking at these drug targets, these causal markers represent putative drug targets. And so I can't help to think that, and when I say I can't help to think, the field has not brought an AI-derived signal to market yet. We're getting closer, but what I'm thinking is that because of the causal, we're modeling the causal nature of human biology at the molecular level, is that the drugs that are produced from this approach are going to be much, much more efficacious.
Yeah.
There's going to be less side effects. We're going to find common signals across multiple disorders. We were talking about this earlier today, looking at common signatures of inflammation. One of the few biological processes that drive multiple human diseases. We find common signatures associated with that. We're going to find causal drivers of these signatures. And wouldn't it be great? And I don't think this is a pipe dream, is to be able to derive or to build a small molecule or a therapeutic strategy that can address multiple disease states simultaneously with far less side effects, because we're going after the causal mechanism that leads to disease. And that's, we're not there yet, but we're getting much, much closer. And all of my early work and what we have published in the literature indicates that we are on the right track. And I'm very excited with working with Dan and his team here, because we have exclusive access to this data. And we have the experts there at the Lieber Institute that can tell us whether that AI is on target or not.
Excellent. Thank you, Tom. And we are sort of running up to our typical 45-minute time window for these events. Just, I guess, in summary, are there any final remarks either of you gentlemen would like to leave with the audience today?
Well, I had mentioned this earlier, Paul. Thank you for the opportunity. Dan, it's always a pleasure when I get to spend a little bit of quality time with you. So I've enjoyed the conversation here this afternoon.
I feel the same way. I mean, we're very excited about the potential here.
Excellent. Thank you, gentlemen. We agree. There's tremendous potential. And for our audience, if you do have further questions or would like more information about BullFrog AI, please reach out to us here at RedChip. You can call us at 1-800-REDCHIP. That's 1-800-733-2447. Or you can also email us at bfRG@redchip.com. It's simply the ticker symbol bfRG@redchip.com. You can also visit our website. We have an information page there for BullFrog at redchip.com/stocks/BFRG. And the latest investor presentations there, fact sheet. You can also even sign up for news alerts on BullFrog so you can stay abreast of the latest developments. But with that, I want to thank our audience for their time today. And again, thank you, Tom and Dr. Weinberger, for sharing the story. We look forward to having you back to share more exciting developments.
Pleasure.
Thank you. Thank you.